mentioned this once before. We're going to start little and go big. We did that with the basic stuff, working away from atoms up to bigger and bigger molecules. When we get into the body systems, we're going to start with the ones that work mostly on the molecular level, things that kind of work bigger and bigger. We're going to do that with the disease organisms, the things that cause disease as well, starting very small and going larger. I'm going to split the videos up into the different groupings. I'm going to put two groups together in this one because one of them we just mentioned before and there isn't a heck of a lot to talk about. And they really are not something you really need to worry about. And those are prions. When we talked about proteins, and I talked about how protein molecules get their function from their shape. They interact with the world with particular shapes. So the proteins that are in your cells are doing all sorts of things. So they have dents that things fit in, and they grab stuff and move it, and they do all sorts of things. A prion is a protein zombie. It is a protein that has the same sequence of amino acids as one of yours, but it is capable of grabbing your functioning protein with the right shape and changing it into a prion, which is the wrong shape for what that protein was doing in the first place. And if it does that into enough of your proteins, whatever that protein was doing doesn't happen. And this causes things like mad cow disease. There's, so far as we know, very, very few human diseases that are caused by prions. And most of them are, mad cow disease is a variant of what they call Creutzfeldt-Jakob syndrome. And there's a few different variants, some of them pretty quick, like mad cow disease, some of them classic Creutzfeldt-Jakob, which takes decades to build up to the point where you have symptoms. So it's possible, this is a fairly new discovery, it's possible that we're going to discover there's a lot more of these working than we thought there were, but it's a kind of a bizarre function, and they're not organisms, they're just molecules. And that means that the same problem you have when you're designing drugs, getting things into the cell where they can be functional, you wind up, any kind of potential prion would need to be able to do that. It would need its delivery system, it would need to get into hosts, it would need to get into cells, it would need to be able to get at the proteins that it could change the shape of. And that's a long shot. It's kind of amazing that these things even exist. So it's not that amazing that there's not a lot of them that affect human health. There's a bad one for bees, but we're not bees. So we're going to come up in size to viruses. But viruses are small. Is that if a cell is the size of the county, then a virus might be the size of a big ocean-going boat. You know, pretty big, but nowhere near the size of something like the county. And I'm not really sure that's a great comparison, but remember, cells are really small. Viruses are way, way, way smaller. You can see cells through a microscope like we have in the lab. You cannot see viruses. When we do our lab where you look at these things through our microscopes, we will talk about viruses because we can do that for the questions, but you can't draw a picture of them, which is something you're supposed to do for that particular lab because you can't see them. Everything else you can see through the microscope. Bacteria are difficult, but you can see them. So, and don't confuse bacteria and viruses. People do that all the time. They are very, very different. So I'm going to talk about viruses in general, and then I'm going to talk about a couple that are major human viruses that act a little bit differently than your typical virus. A classic virus is a casing, usually made out of a collection of proteins, sometimes a piece of host membrane that it steals and takes with it, and it's got DNA inside. Now there are variants. is a lot of times there's DNA and there is maybe one or two other proteins that are brought with it in order to help it start up in the next host cell. But these things need to get into a cell in order to do anything. So something on the casing is going to hit the host cell, stick there, and then changes will happen that will move at least the DNA into the cell. Sometimes viruses, the whole viruses get into the cell and then the casing opens up. A lot of times the virus opens up a hole and the DNA goes through, and if there's something with the DNA, it goes in also. Because really, it's the code that needs to get in there. So these things live in cells. When we're trying to fight things, we are trying to fight these disease organisms. We're trying to poison them, duh. But the trick is we need to poison them without poisoning us, without poisoning the host. Now, it'd be nice if we could also kill them between hosts. And that is not that easy to do. Now, it's even harder to do with viruses because a virus between hosts isn't doing anything chemically. It's got that contact thing that will react with the host cell. But other than that, they're just kind of floating around. And being very, very small is that they have many, many ways to move around. Different viruses move in different ways. COVID, SARS coronavirus 2, gets expelled in respiratory droplets. How big the droplets need to be carrying viruses, they're still working out. Flu is spread that way also. Ebola is spread in body secretions. It actually makes you bleed. You can pick it up from contact with blood and other sorts of things. Herpes causes an eruption on the surface and viruses are shed onto the next person that way. But they got to get to the right kind of host cells and make contact with it. I'm going to talk about COVID a lot because we're in the middle of the pandemic. COVID works a little bit differently than some things, but they do have on the surface, you see the pictures of COVID, it's always got these spiky things sticking out of it. Those contact a particular molecule we have on our respiratory cells, also other places in our body, and it could, viruses that get out of your respiration system and go other places in your body may be doing some pretty nasty things. And again, still working that out. But that's how they get their RNA and at least one protein that it carries with it into your cells. So it gets in there. Now here's a very important distinction for viruses. Once they're in your cells, they co-opt a whole bunch of things that your cells normally do. Basically, your protein making chemistry to make virus proteins. We can't poison that without poisoning that same system all over your body. Your body cells are making proteins for what they do all the time. So there isn't really a lot in these stages that we can do. Your body has ways of recognizing a cell that's been taken over. It has some ways of recognizing when there are viruses out and about, and that's your early warning system that fights them. One of the things that COVID does is it shuts down a very important part of that. We'll talk about immunity later. But once COVID is inside our cells, it's taking its RNA, which is part of how the coding system works, and feeding it through your protein making machinery and making proteins. It does one odd thing where the protein that it carried in with it is kind of watching this long string of amino acids coming out. Now typically you'd make a protein and that'd be it. And then you'd use a different part of the code to make a protein, and that'd be it. You'd use a different part to make a protein. Is that the one that it brings in actually snips the strings in order to cut, so the proteins are kind of continuous. And I would assume the first thing it makes is more of the snipping protein, but it's an odd system. Now, for most viruses, they're also making DNA. COVID's making more RNA, because it's got to package more RNA in every virus that it releases. When we make copies of our codes, mistakes get made, but we actually have a whole bunch of chemicals at play trying to keep the system from making mistakes. Basically, reading the code as it's copied and going, whoa, wait a minute, this is bad, back up and try it again. And it's called proofreading, just what it sounds like. It makes sure that the spelling of the code is right. A lot of viruses use your system to make more DNA, but they don't use the proofreaders, which means you get more mutants. Mutants produce variety. Variety is a fairly strong aid in evolution. Just as I said before, most of the mutants are not going to do anything different. Is that there's a few mutants of COVID wandering around. One of them does seem to be slightly more contagious, but most of the other ones, it doesn't seem to have changed anything about anything. And you're going to get a lot of mutants that don't work because you've produced a code and the protein that it makes just doesn't do what it's supposed to do. Those are kind of acceptable losses in an evolutionary arms race, which is what's going on between the host and the disease organism. Also, while I'm on the subject, a lot of people don't consider viruses to be alive, so they wouldn't necessarily be called organisms. We're ignoring that. So they're making more viruses. Now the escape mutant is something they specifically came up with as a term for HIV that causes HIV/AIDS. HIV, again, another weird virus. Remember I told you I'd talk about a couple of viruses that don't follow the rules exactly. HIV is an RNA virus. It carries RNA in it, in a protein casing, actually kind of a cylindrical protein casing. And it's got a couple of enzymes, a couple of proteins that it brings with it. And those proteins it can use to take its RNA and make DNA from the RNA. And then make more and more and more DNA, and then it sets your system in play to make proteins to make more viruses. And eventually it makes more RNA to package in the viruses. It does not use our proofreading machinery to make its own DNA. So it produces lots and lots and lots of mutants. You take in HIV, it goes into a player in your immune system, one particular type of white blood cell that if your immune system is an army, it's like the middle management, it's like the sergeants. It's not doing that much fighting, but it's telling everybody who's doing the fighting what to do. So it's a really important coordinator cell in your immune system. And HIV is breaking them open to release more viruses. So this HIV in those cells does kill them. Here's the thing though. Your immune system, pretty good, is that HIV doesn't act that quickly. When the viruses get into your system, you start to make antibodies to it. And before very long, when HIV is released and trying to get from one of your immune cells that it just killed into another one to make more, and then it gets into fluids and gets carried to the next person. But it's trying to spread in you too. It's trying to build up its population in you. And it's kind of hiding in your immune system. But once you have antibodies to it, you're actually pretty good at keeping it under control. There's a lot of people that are HIV positive for a very, very long time, And they don't progress to AIDS, which is the syndrome you get when so many of your Sargent cells, your T4 cells, are wiped out that your immune system ceases to function. It doesn't work right. And then you wind up with immunodeficiency, the I part of HIV. Here's what you don't want to have happen if you're HIV positive. You don't want a mutation that has two abilities. Number one, it works. It's a working virus. And two, whatever molecules your antibody's attached to, it's changed shape so that they don't attach to it. So it's become invisible to the antibodies that you've made. You will register that and set off to make antibodies to this new one. But in a lot of people, by the time the antibodies are out, the Sargent cells are gone and the immune system doesn't work anymore. So having antibodies is useless. The antibody's got nothing to signal. And then you go to AIDS where you're immunodeficient and you can't fight anything off. So that's what an escape mutant is. You do have a handout that talks about this a little bit, and it is one of the big deals of HIV. The viruses need to get out of the cell. And remember, it's really hard to treat them when they're active and they're making viruses because they're using your machinery to do it. Some viruses have a very particular way of getting out of our cells that doesn't match anything our cells normally do. And that's an area that we try to attack them at. Keep them trapped. Don't let the viruses get out. Tamiflu does that, where the particular proteins inside the membrane that are at play to get the viruses out, the flu viruses out, Tamiflu blocks them. It doesn't do a great job. If you get it really early, it can keep them from spreading enough viruses to you to build up an infectious, symptomatic, or more symptomatic bout of the flu. But once the viral population has gotten really built up, Tamiflu isn't that useful anymore. I forget what the drug is that they use on herpes viruses. Herpes viruses work the same way, where it goes after a protein that allows the viruses to get out of the cells. And so it reduces your own immune response to it, so it tamps down these symptoms and it makes you less contagious. But that's rare. Very often we don't have a way to attack a virus when it's active because there's nothing it's doing that we can afford to poison, or we can't get at the molecules that are different. And once it's out of your cells, really, we can spray disinfectant on a surface and make the viruses fall apart. Some viruses are very hardy out in the environment. Some of them require certain types of conditions. We're just about to head into flu season. Why is flu season during the winter? It's because flu viruses are very resistant to drying. Some viruses aren't. HIV, if you dry it out, it falls apart. But flu viruses are very resistant to drying, but they like very stable kind of indoor temperatures. So they're an indoor virus. When you get into the winter, the air gets dry, it's pretty controlled in terms of temperature. So flu likes to pass from person to person inside these kinds of environments. It was always crazy to think that coronavirus was going to get worse during the fall because it obviously could spread in hot and humid, even outdoor conditions, although it's harder to get it from person to person in outdoor conditions. And again, if you pass it in respiratory droplets, then the farther the respiratory droplets can get out of you, the more they can spread to other people. The 6-foot rule tends to kind of isolate your little bubble, because they are small, but they do drop. Maybe one of the reasons why, nah, I'm not gonna say that. Anyway, but having something that pushes them out the side of a mask, right? You're wearing a mask and you go, well, the air's coming out anyway. Yeah, but you've slowed it down. You've bounced it off the surface, which actually catches a lot of your respiratory droplets, so a lot of viruses don't get into the air. And it does make your bubble of infection smaller. And your ability to pull things, you're breathing around the edges of the mask more than through the mask. So it is more effective at keeping you from spreading it than it is at keeping you from picking it up. But it's effective at blocking virus coming in as well, because it's got to get around the edges of everything. And the viruses, you know, are sticky. Is that they make contact with cells in order to find the right molecules, so they do tend to stick to surfaces if it's kind of a biological surface like your skin or even the cloth of a mask. So as much as the masks have become this weird political thing, from a public health standpoint, it's a really, you know, they're effective. And they're effective in a very commonsensical way. You don't necessarily, it's not, I mean, didn't the explanation I make perfect sense? You don't need to be a trained biologist to understand that. Anyway, so viruses are, they're mutating. That sometimes makes them able to move from one type of animal that they've been very happily working in into another type of animal that can support them. Flu viruses do that all the time, where humans spread them to pigs. They work in the pigs for a while, and then a variant comes out that is a different variant, but now can infect humans. If it's the same variant that comes back, you know, we're already dealing with it. I like to talk about flus specifically for vaccinations, because that's a vaccine that's out every year. And you'll hear people go, oh no, it's not, it doesn't really work. It's like 50% effective. Here's what happens with the flu vaccines. Right now, I'm making this in August. Somewhere in the world, a bunch of experts got together back in March and looked at the state of influenza and what the various variants were, this is what they call the strains that were around. And they went, okay, when we get around to October and November, what are people gonna be dealing with? And they select 3 or 4 of them, trivalent, quadrivalent vaccines. And they go, okay, these are the variants that we're gonna package into the flu vaccine, because it takes months to produce this stuff. And so right now in August, somewhere in the world, in several places, they're making flu vaccines against 4 flus that they expect to be the big players in the fall. Sometimes they guess right, and it's really useful. The 50% year, which was a few years ago, half of the flu variants that were around that year weren't in the vaccine. So it worked against the half that it worked against, but it didn't work against the half they didn't expect. So that's what it means, is that it's, you know, it's an epidemiological guessing game. And, you know, and they're good at it, but you can't always plan for the arisal of a new strain. I'm sure, well actually, there's a new strain I read about like 3 weeks ago in China, coming out of chickens. It wouldn't have been on the radar in March. Now maybe nothing will ever come of it. Or it may be a huge player in November. Is that even in August, there's no way of knowing. So it's a very tricky thing. It's better to have a virus that's fairly stable. And I should mention that COVID has some proofreading capability. It doesn't make as many mistakes. It doesn't produce as many mutants. It doesn't produce as many variants. So if we make a vaccine to it, there's a very good chance that it will work for a long time. How long we're immune, they're still guessing at. One indication is at least 17 years. That's a pretty good time. But also it turns out that if you've been exposed to other coronaviruses, you've made antibodies to some molecules on the virus that match the new one, the COVID-19, and not enough to keep it from completely setting up shop in you, the way it would for viruses specifically for COVID-19, but enough to reduce the amount of viruses that can get around from your cell to your cell to your cell to give you symptoms. So they think that people who've had some of the few coronaviruses that cause colds, people that work in veterinary offices where they're actually exposed to cat coronaviruses, that they may have antibodies that are protecting them from getting really, really sick. One of the things that seems to protect us from being really, really sick. Resistance, remember resistance? Okay, that's it for prions and viruses.